our next speaker for the day will be Dr. Fiona Malone. She is a biomedical engineer, she is a lecturer, a science communicator, a comedian, as well as a dog mom. No stranger to competitive science communication, she was the 2015 national winner of Thesis in 3, as well as the 2019 FameLab Ireland runner-up. She has also represented the Galway Mayo Institute of Technology at other science communications events such as Inspire Fest's Research 2016, as well as Soapbox Science in 2017. Today, she will chat about biosimulators and how they are created from translating medical data into engineering data and to fabricating patient-specific anatomical models, which can be used for medical device testing and analysis. Over to you, Fiona.
Thank you so much. Hi, everyone. How are you doing? And kudos for making it this far. Good job to everyone for speaking this evening, and a massive thank you to everyone for organizing it. I wish I was in a bar right now, but unfortunately, and I can't help you with this quiz. I'm not a Corkonian. I don't know what this is, so no one can bribe me. I don't know where I'm supposed to be right now, but I'm here anyway in the west of Ireland chatting to you. So I'm just going to share my screen and try something a little bit different this evening with Mentimeter. I'm not sure if people are familiar with it, but we'll try and get a bit of interactivity going on with this evening. So thank you for that introduction. I am an engineer, a lecturer, science communicator, comedian, and a dog mom as of late. I'd say a no-time comedian at the moment, seeing as there are no gigs going on, but I am a full-time dog mom, which does have its challenges. And it sparked a bit of interest on Twitter as we were getting ready for Pint of Science that would the dog be here this evening. He's not physically here, but this is Reggie, much more interesting than I or my research. And it's probably why 90% of the people have come to listen to me this evening. So this is Sir Reginald M. McHugh, first of his name, potentially the rightful speaker at this evening's event, destroyer of socks, barrier of bones, and breaker of hearts. So that is my little dog fur baby there. For the other 10% of you that have turned up this evening to actually listen to my science, I'm going to chat about biosimulators, the advantages of them, how they're made, which is super cool, and then a bit of a case study on what I've done in the past with biosimulators. And I'm so thrilled that Matteo has brought up soft robotics, because that's just made my whole talk so much easier, which is great. So if you can, go to www.menti.com, and using this code, you might let me know when you hear the word biosimulator, what comes to mind. Maybe you've never heard of it before and something might pop into your head. So let me know, and the ideas and suggestions should pop up on this screen now. Hopefully everything will work out okay. So the Matrix. Some of these are great already. And like Matteo, I often get thrown at Terminator, Big Hero 6, all of those guys, Cyborg from the DC universe, and all of those tend to pop up here. So humanoid, very cool. Oculus Rift, amazing. So I'll give it a couple more seconds on that, not to waste too much time on this, just to see where everybody is at on the biosimulator sphere. So bioreactor, very cool, yeah. Virtual organs, getting a little bit closer to what I'm actually going to talk about this evening. So anybody that's here to listen about the Matrix, you might be disappointed. I'll just give it another second or two and see is anyone else going to pop in. 3D body models. Oh, that might be my mom. Told her what I was talking about tonight. Super. So I might move on just in the interest of time, and thank you for everyone for indulging me in that there. So sometimes when we think of biosimulators, we might think of computational models. I'll just move that out of the way there. Such as this model here of a musculoskeletal system of the female anatomy. So that's just the muscles and the bones that comprise the body. And computational models are super helpful because they can give us a lot of information and predict a lot of information, especially like what Niall was saying earlier with the prediction of COVID-19 expulsions from the mouth and nose. But they're really helpful when we put them together with robotic information, like what Dr. Matteo was talking about in this. I love this video from the lads over at MIT of trying to analyze human gait analysis. Kind of looks like me the morning after a Pint of Science event. Maybe not this year though, unfortunately, or fortunately, maybe my liver is probably delighted with that actually. And as we can see, they're comparing the physical robot with their computational model to a really good effect. And this is where we have some really interesting data coming up. As Matteo was saying, this kind of robotics isn't indicative of what's going on with our human anatomy. So over the last 20 years, we've been looking to animals and nature as inspiration for more realistic, I suppose, biosimulators and biorobotics. Now, I don't know what animal they're looking at here, but my dog definitely cannot do this because I have tried to make him do this. So instead of looking at these kind of rigid fixtures and really tough electronics and tough materials, we're looking towards a softer side of robotics that I'm absolutely obsessed with, which is soft robotics. Now, I know Matteo was saying he's an octopus aficionado, but I'm more of a starfish kind of gal. And that's a terrible joke. But anyway, so this is the soft robotics, the idea where we would inflate and deflate parts of our silicone or rubber-based model that would change the direction and movement of what we needed to do. And this is where they come into play in the biosimulator world. So the advantages of biosimulators, sure, they're great, but the first one is that they can help us test medical devices. And these are really important, especially in the medical device world, in order for us to test medical devices and optimize their performance. So you don't have an issue like these surgeons in the clinical setting here. You might have a medical device company, for example, working with catheters and balloons and stents, and they want to optimize their performance in maybe an in vitro silicon tube as opposed to testing it on an artery or a vein. And using the results of these designs and these test results, we can quicken the design process. So the biosimulators are really helpful for especially medical device startups where the funding for animal testing and things might not be there initially, but some excellent results can be achieved using the silicone and rubber models. And while we chat about animal trials, if you're working in the medical device sector, animal trials can be an important milestone to achieve and to pass in order to get your device onto the market. However, having a successful animal trial is particularly difficult to achieve. So biosimulators allow medical device companies to ensure that their device is testing at the optimum level to achieve a potentially more a better result, I suppose, in terms of the animal trial. And I know we're here for Tech for Humanities, and it would be an absolute dream of mine to see that maybe biosimulators will become the gold standard for preclinical testing, thus reducing the need for animal trials. But that might be a pipe dream. We can also simulate disease in biosimulators and focus specifically on a disease state, which can often not be replicated in animal models. So for example, if you're working in a device company that treated aneurysms, maybe aneurysms in your tummy or in your brain, this might be difficult to replicate in an animal model, but in a biosimulator using different materials, a patient-specific model could be created. So that specific patient with those specific blood vessels and that actual aneurysm could be replicated in order for the surgeon or the clinician to decide what they're going to do with the surgery, which I think is really cool. Training methods are also really helpful, an aspect of biosimulators. So anybody in the medical field, doctors, surgeons, nurses, physios, they can all benefit from different types of training methods, and biosimulators can offer an alternative apparatus in their training situations and scenarios. So I suppose what some people might be here to listen to is, how do I make these? These look cool and all, but how do we get there? So we have a look at specific types of medical data, so we can convert these images into 3D printed models, and obviously then into much more functional models for our biosimulators. The medical data we're looking for is things like CT scans, MRIs, fluoroscopes, images, X-rays, and ultrasounds. Obviously, different images can behold different results. An ultrasound can be a little bit difficult sometimes to visualize what is going on, and certain images and characteristics wouldn't be included, whereas a CT scan, you've got all of the different layers going on and we can really see what's inside you, which is great. And then using different softwares, we can recreate three-dimensional models. So we can analyze them not only as a computer biosimulator, as we saw earlier, but we can bring them into the three-dimensional real world as well. Certain softwares that I could recommend would be 3D Slicer as an open source software, or Mimics is another really good software that your college or your university or your research institute may have a license for. We also need engineering data for our physiological simulations. So this kind of data is your flow rates of your blood flow rates, your blood pressure, anything that can give us information to create the entire scenario and the physical simulation for your biosimulator. And this is the real information that we need in order to create it as a patient specific. So as we look at my case study in a few moments, we will have a look at the different types of information I would need to make each scenario different. We need both of these to build clinically relevant biosimulators, and they're as important as each other. Okay, so you're never as good unless you have the quality of tools that you have, and these are what we need to make these biosimulators. So I hope I'm not doing too bad on time, but just to recap, biosimulators can take many forms, shapes, and sizes, and they can be patient specific. They are helpful for medical device design and testing, and can reduce the number of animal trials required, and possibly in the future someday, maybe reduce them altogether. I do that for Reggie. Useful training apparatus for medical staff and students, and they can also be useful in understanding specific disease states, which is what I'm going to look at now in terms of my research. So this all sounds pretty straightforward, right? I mean, I know I feel like I'm rushing through it, but hopefully everyone is on board with me this evening. So we're going to have a look at a case study in terms of my research using biosimulators, and the type of research I was looking at was a disease state, okay, and a specific disease that I was looking at is stroke and atrial fibrillation. So just a bit of background. A stroke occurs when there's a lack of blood flow to the brain, which obviously is carrying oxygen. This can result from two different reasons. We could have a burst blood vessel in the brain causing a hemorrhage, or a hemorrhagic stroke, or we could have a blockage. And these blockages can be caused by lots of different things, but the blockages that I'm particularly interested in are blood clots. Okay, and a major contributor to blood clots within our body and blood clot formation is atrial fibrillation. So hopefully this video will work there now. So in atrial fibrillation, oh, I clicked out of it, sure, of course I did. In atrial fibrillation, what happens is your left atrium beats out of sync with the rest of your heart, causing blood to pool. And when blood pools, it clots. And so in a single heartbeat, a blood clot, which is denoted by this little black spot here, can be emitted into the cardiovascular system, so into your body. And the real scary scenario occurs is if that blood clot travels to your head and blocks up the tiny vessels within your brain and causes a stroke. And that's where we have the serious issue. And what I was really interested in is why this happens. So what I wanted to look at using my biosimulator was why some clots travel to the head and some don't, and what various scenarios affect that. So is it the flow rate? So is it a healthy flow rate, or does this atrial fibrillation really contribute significantly to a stroke? The different types of clots that you might get within your system, and how big or how small these are formed within your heart, and also the number of clots. Is it fair to say that one clot might be produced in your whole lifetime, or would lots of different clots occur at the same time? So that's a really interesting way of looking at the stroke occurrence. So going back to what we were looking at earlier, so the medical data that I needed were CT scans from patients focusing specifically on their aortic arches.